Good morning. I'm Hazim K. Ekenel. I'm a professor of computer engineering in Istanbul Technical University. I am mainly focusing on computer vision and machine learning, actually. And this presentation was intended to be a recent literature review in the developments of insect classification in computer vision, but we had to squeeze it down. So I will talk about very, very recent advances in the field. The presentation is prepared by my graduate student, Elif. So actually, we mainly analyze humans, not insects, in our research lab. So we have worked a lot on face recognition, human behavior analysis, and recently we're also paying attention the problems in this domain. So we had this deep learning revolution, and many people use these backbone models like models pre-trained on ImageNet, like the ResNet, AlexNet, GoogleNet, etc. And this has been the main thing so far. But in recent years, we had also seen some foundation models and even some large language models focused on insects. The main problem we noticed in the field was about the utilization of microfeatures between different insects, because you have many different classes and you need these microfeatures to correctly identify the insects. Therefore, the recent methods focused more on learning discriminative features. So what are the challenges we are facing from the computer vision perspective? There are 5.5 million different insect species, high intra-class and low inter-class variances, so it's difficult to model each class and discriminate them from each other. They have different forms at different growing stages, and then recently there was this talk on detection. You might also have this background clutter, so it might be difficult to discriminate the insect from the background. And the data sets we have been seeing, that are publicly available in the computer vision field are since till 2023 is like that. And as you can see, over the years, the number of classes have increased, as well as the number of images. Now we have Insect 1 million data set also, where they collected around 1 million images of insects with some description information. So this includes, for example, genus, subphylum, order, class. In terms of the representation, now the recent models are more focusing on matching the natural descriptions, NLP descriptions with the image. Before that, there are also two recent data sets, but they are more like macroscopic imaging. One is Bioscan 1M. These are high quality microscope images of 8 taxonomic levels. And another problem in the domain is class imbalance. We also had a talk on that. And then they also recently have this Bioscan 5M. So besides the imaging data, they also benefit from the DNA sequences. And in terms of representation, the recent representations are more based on clip-oriented methods. So these are very good for zero-shot or fifth-shot learning. So the generalization strength is relatively high. So you have the images plus the descriptions that you learn. This is what they have. BioClip, so it was a pre-trained clip on Tree of Life 10M. And then later on, they also had this CLIBD that also utilized the DNA sequences in the learning process. So briefly about the insect foundation model. So the recent trend is now more like having a foundation model initiated with this insect foundation. This was presented last year at CVPR. And then the same group also has a large language model based on Lava. They haven't released the models yet, but soon it will be available. So what they benefit from is that masked autoencoders. So this is an advanced representation approach within computer vision. And then also vision transformers. So since we have more data, they tend to move the efforts towards the vision transformers instead of convolutional neural networks. And then the points that we are interested in at the moment is how to handle the imbalanced data problem. Also, like these are generally bounding boxes, how to benefit from the LLMs for also descriptions, like interpret the results we have, as well as maybe some more focused low data regime problems, because again, these more recent models are more powerful in terms of generalization capability. Thank you very much. We'll bring all speakers up.
Okay, thank you. That actually concludes the first session of presentations. And I'd like to ask all the speakers to come up here to answer any questions from the audience. So thanks to all the speakers. We actually fought up a little bit, so we still have time for questions. So I'll just pass on the microphone and yeah, please, if you have questions, I don't know, maybe maybe you can get microphone if someone has a question. So yeah. Yeah, go ahead.
Thank you for your presentations. I have a question about the millimeter wave data. Your study so far has been in the laboratory, I understand it. In the fields, what's the sort of detection range of these systems, and are they expensive systems to deploy?
Maybe, yeah.
One of these?
Can we turn on one of those? And maybe you can.
It should be on there.
So here we are mainly intending near field applications. So like in one 15-meter kind of distance. So we will be identifying the insects that are flying closely to the millimeter wave antenna. We would also planning to do some setups so that an insect could pass near to the millimeter wave antenna and pass through it like that. Yeah.
Another question, have you seen like individual signals in your sound patterns? Because I'm imagining, well, maybe not for a bee, but for maybe like butterfly, you can have like really small generic, but with some unique in there, for example, that could also create one kind of sound transfer. So would it be potentially in the future be possible to identify individuals, or maybe not individuals, but the age of insects they are on that?
So age of things, sorry?
So the age of the insect, if it's like flying off, because it's the butterfly, the scales, the or like it's timbers and something like that. Imagine I'm not familiar with these, but like similar things could happen with these.
So the activity of the insect in the different stages, that is a good question that has to be studied at different time period and different life stages. Generally, based on their muscle cycles and their size, there is certain range of wing of flapping frequencies for each types of insect. For the moment, butterfly, it has a larger wing size and small body, so wing flapping frequency will be much lesser compared to the bees. Like that, every species would be has certain range of lower and higher wing frequency ranges, and that might be changed based on their activity and different stages of life that has to be explored more.
Very interesting talk. And I was wondering whether the clutter effects, the movements have an effect on the signature, and maybe the position of the insect will also have an effect on the signature. Can you define that when you create your reference dataset or you have page signature, are you able to cover all the orientations and movement that you
Actually, orientation doesn't affect much on this micro Doppler effect because the system can only see the changes in the motion. So also in the setup, insect is freely moving in the box. So different orientation also, it's creating same Doppler effect.
It's so I'm very excited about clutter, but I am also very anxious and acceleration because of the quality of the data that we have in GBIF. And we know that many of those data were not validated by other expertise, and so we have a high percentage of misidentifications. And so if we do a scraper and get directly automate this in models, then we are adding, which is happening already with many models, we're adding error. Do you have a way to take that into account with your model system?
Yeah, actually, it's on the they say taxonomic level. We don't have like a label in GBIF that would say this is a very high quality label, so it's validated. But we also have we are designing our image cleaner model because actually we found out that almost 10% of the image that we downloaded are either noisy or it's just a background, just leaf, or it's just it can be also microscopy images of just a part of the insects. And we are designing now a tool, a deep learning system that just sorts valid insects or adults, for example, from the rest of the images. But yeah, I think it would be an open question to GBIF themselves if they can have a tick. One of the metadata could be expert validation. Yeah.
This is a good question. So it was because the species was somehow only active very late in the season, and that's why the model predicted it to be active at colder temperatures. And we don't really know because it was like this, because the species technically would also occur in midsummer, but we could imagine that it might be because the cameras were faced on sedum flowers, and it was a mix, and maybe so some of them have been flowering later in the season. So it could be that this hoverfly species was specialist for these flowers, which are flowering later in the season. That's why everything got messed up a bit. So, but yeah, in the end, we don't know.
Question for you. So it's hard to figure out the type. I thought I saw something like a negative effect of the time chemicals on the butterfly presentation compared to the control. I could be wrong about that, but I thought I saw something like that. But in general, can you comment on the possibility of using your system to look at competition between different types of pollen mixes for a given resource?
So we didn't test for the time effect, so I don't know where you find this time.
Sorry, times the.
Yeah. Okay, sorry. I know. Actually, this is a good question. So because we were early in the season, so from May to June, it's actually the flowering time for the Buddleia species, and the timers come later on. So that could explain why the timer solution was not attractive at the early season.
Attractive to bees.
But it was attractive to bees also because every time we had the thymus without nectars and with nectar. So there is something that we have to figure out if it's only the nectar that attract bees or if there is also chemical compounds